Hey guys, so in Cell and Moleque, I know we all have become quite familiar with our cloning projects. Most of us are experiencing the frustration that has come with trying to isolate our gene through PCR and cut it properly for the ligation reaction. Well, I have had the opportunity in the research lab to work with a different cloning mechanism known as TOPO cloning. So today I want to review the standard method of cloning and introduce you guys to this newer cloning process. So what is TOPO cloning? It's a new cloning mechanism, and it refers to the process of cloning a gene into a vector without the use of restriction enzymes or DNA ligases. So first, let's review the method of standard cloning. First, you need to obtain your gene of interest. Now, your gene can come from a plasmid, referring to a circular piece of DNA, or it can be isolated from a tissue sample. For our purposes in lab, we obtained our genes of interest from fly tissue samples. First, we isolated RNA from this tissue to obtain the expressed version of our genes, free of untranslated introns. Then we made cDNA from this RNA so that we could amplify our gene via PCR. Primers were designed to isolate our gene of interest, but included on the primers were the restriction sites for restriction enzymes that generate sticky ends that could be recombined with the vector once it was cut with those same restriction enzymes. The restriction enzymes chosen were those that did not cut within the gene of interest, but they were part of the multiple cloning site on the vector. They had to be two different enzymes to help ensure that the gene would be ligated into the vector in the proper orientation. Once the gene was ligated into the PUC19 vector, a series of bacterial transformations and patch plate screening was completed to identify bacteria colonies that took up the vector containing the gene of interest. The positive colonies were then used to begin overnight growths that would be mini-prepped and sent for sequencing to confirm presence of the correct orientation of our gene of interest. The TOPO cloning process is very similar to the standard method of cloning that I had just described. However, it differs in the design of the primers to the gene of interest. Instead of designing primers with restriction enzyme recognition sequences, the forward primer is designed with the TOPO recognition sequence. The primer has the sequence CACC added to it to create the TOPO addition to the gene that can recombine with the P-enter vector at the recognition sequence sites without the use of a ligation reaction. Furthermore, the PCR clone of the gene with the TOPO sequence addition must be replicated with a polymerase that produces blunt ends. Once the gene of interest is inserted into the TOPO P-enter vector, the gene can easily be flipped into other expression vectors. So why would we use TOPO cloning over standard cloning procedures? TOPO cloning comes in a kit with all the necessary reagents in one isolated location. This kit itself is advertised as the fastest, simplest, and most efficient cloning kit available. The reaction takes place in about 5 minutes and results in about 95% efficiency, meaning that 95% of your colonies should contain the plasmid with your insert in the proper orientation. TOPO cloning is also said to be a flexible cloning process. There are 3 different kits for TOPO cloning, depending on what type of gene you are cloning. The process I described corresponds to the high-fidelity blunt-end TOPO cloning. However, if you have an exceptionally long PCR product, there is a long fragment cloning process available, and there is a kit referred to as TOPO TA cloning for subcloning and in vivo transcription processes of your gene of interest. TOPO cloning has also eliminated the blue-white colony screening process in standard cloning methods because only the colonies containing the plasmid grow. TOPO cloning also offers flexibility in what type of vector your gene of interest is ultimately cloned into based on the purpose behind your cloning reaction, whether it be for sequencing or subcloning purposes. The main reason that we have done TOPO cloning in the research lab is because of its versatility when it comes to the variety of expression vectors that the gene can be flipped out into. In the research lab, we plan to utilize the gateway cloning reaction to flip the gene of interest out of the P-enter vector and into a few different expression vectors without having to go through the restriction digest ligation process for each vector switch. For gateway cloning, a single recombination reaction can move a piece of DNA from one vector to another. As just mentioned, this process is simpler than standard procedures as it eliminates the use of restriction enzymes and the ligation reactions. This cloning process assumes your gene is cloned into a donor plasmid, which was completed by the TOPO cloning method as just described. Then it utilizes the recombination sites from the donor plasmid to complete the reaction and transfer the DNA into a series of expression vectors in which your gene of interest can then be expressed in the desired cells or tissues. However, what are the disadvantages to using the TOPO cloning method? For one, it is much more expensive. The TOPO cloning kit costs hundreds of dollars and only contains 15 to 20 reactions. Also, a lot of time and preparation needs to be taken into consideration when deciding on the proper kit to purchase and proper vectors in which to clone your gene into based on all the possible purposes for your cloning reaction. Furthermore, this cloning method is not foolproof. The P enteroplasmid is not a high copying plasmid, meaning that many prep concentrations after overnight growths of transformed bacteria are relatively low. Essentially, even with this new and improved method of cloning, nothing works perfectly, and I am experiencing many of the same frustrations as you all have been experiencing in lab with the standard methods of cloning. Overall, TOPO cloning offers a new avenue with which to complete cloning reactions and insert a gene of interest into a plasmid vector. Differing from standard cloning via the primer design, TOPO cloning follows much of the same procedure as standard cloning methods. I hope this presentation introduced you to new cloning methods and expanded upon your laboratory knowledge from your own personal experience and frustrations from using standard cloning in this lab.